so bad at the technical stuff, honestly. Like if you guys watched my previous one, you know मैंने कहा था कि I'm running out of my data, that this blah blah, but trust me, फिर मुझे पता चला कि यार नहीं यार है, मैंने अभी जाके चेक करा. So this is the, मुझे दोनों साथ नहीं करना था, but never mind. आज हम करने वाले हैं plant physiology the same day, by the way. अब I didn't even had like proper 15-minute break also. I just went to बाजू, हमारे घर के बाजू शॉप है, ठीक है. I got this are really my favorite. Please don't call me now. कितना गंदा खा रही है, unhealthy. This is my favorite. दिख रहा है? दिखाती हूँ अच्छे से दिखाती हूँ. This is so yum. Not. I really don't like eating in front of camera, in front of someone, but still. Say hello. No, it's me actually, because I don't like, no, you like seeing, that is another. This video is a welcome back. Let's do plant physiology. Let's start. दो मिनट में I'm on water. I didn't even try water and pakla in my break. Mechanical stuff to steam. Water synthesis, transportation, mechanical. I'll just bring water. Done. And okay. Characteristics of root for absorbing water. The ability of the roots to draw water from the soil is dependent on the three characteristics: a huge surface area, and the huge surface area, root hair containing the solution cells up at a higher concentration than the surrounding water, the root hair having thin wall. That only. Surface area of the root is enormous. Root hair contains the cell sap of the higher concentration than the surrounding water. Root hairs have thin cell wall. Root hairs have thin cell wall. What are the characteristics of the root hair for observing characteristics of roots for observing water? First is what? The surface area should be enormous. The root hairs containing the cell sap should be higher concentration than the surrounding water. The root hairs should have thin cell wall. Next, imbibition. Imbibition is a surface attraction of water. Imbibition is a phenomenon by which the living or the dead plant cells absorb water by surface attraction. They are hydrophilic. Okay, diffusion is the free movement of molecules from a substance, solute solvent, from a region of higher concentration. Diffusion क्या होता है? From a region of higher concentration to a region of lower concentration. From a region of higher concentration to a region of lower concentration when the two are in direct contact. So what is diffusion? Diffusion is a free movement of molecules from the region of higher concentration to a region of lower concentration. From a region of higher concentration to a region of lower concentration. What is that? That is diffusion. When the two substances are in direct contact. What is osmosis? Osmosis. Osmosis is the movement of water molecules from the region of higher concentration to the region of lower concentration. or with a lower concentration through a semi-permeable membrane, that is what osmosis. Now we have endosmosis, we have exosmosis. Endosmosis, we have exosmosis. So what is endosmosis and exosmosis? Let's see. Endosmosis is the movement the inward movement of water through a semi-permeable membrane when the surrounding solution is less concentrated. Exosmosis is the outward movement of the diffusion of the water through a semi-permeable membrane when the surrounding water is more, so the surroundings when the surrounding solution is very concentrated. It causes shrinkage of the cell. ठीक है. नेक्स्ट ये एक्सपेरिमेंट लिटरली मैं स्किप मार रही हूँ, ठीक है यार, नहीं मारेंगे, ठीक है. लास्ट में मैं एक्सपेरिमेंट्स का वो सेपरेट होगा, करेगी तो सच में करेगी, बट आई विल डू सेपरेट, डोंट वरी. What is osmotic pressure? Now, osmotic pressure of a solution is a measure of tendency to take in water by osmosis. Osmotic pressure of a solution is a measure of a tendency to take in water by osmosis. Tonicity, relative concentration of solution that determine the direction and the extent of diffusion is called tonicity. Based on it, the solution can be of three types: isotonic, hypotonic, hypertonic. Isotonic, the relative concentration of water molecules and the solute on the either side of the cell membrane is same. In such a solution, there is no net movement of water molecules across the cell membrane. Hypotonic, in this condition, the solution outside the cell has a lower solute concentration than the fluids inside. As a result, the water molecules from outside will move into the cell, that is endosmosis. Hypertonic, in this condition, the solution outside the cell have a higher solute concentration than the fluids inside the cell. Consequently, the water molecules from the interior of the cell will move out. Exosmosis. Okay? The results may be as follows: cell shape and size remain changed. The solution outside is isotonic. Cell shrinks in size and loses the shape when it is hypertonic. Cell size enlarges or even bursts when it is hypotonic. Okay? Now, in the case of the plant cell, another striking feature determines the behavior of the cell when subjected to varying external fluids environments. This feature is rigidity of the cell wall, which resists bulging and protects the delicate cellular parts inside. The phenomena related to this behavior are turgidity, plasmolysis, and flaccidity. Active transport is a passage of a substance from its lower concentration to the higher concentration using energy from the cell. Okay? Passive transport is nothing different from diffusion. Passive refers to the requiring no input of energy. There is free movement of molecules from their higher concentration to the lower concentration. Without, there is no energy. ठीक है. Turgidity and flaccidity. Every living plant cell, directly or indirectly, is in contact with fluids. The root hairs in particular are surrounded by a soil water. All such cells are subjected to osmosis and the water continues to enter as long as the cell sap is more concentrated than the surrounding fluids. When a cell reaches a state where it cannot accommodate any more water in it, fully distant, it is called turgid, and the condition is called as turgidity. When a cell is turgid, its wall is stretched under pressure from inside, and in its turn, it presses the cell contents out towards the center of the cell. The pressure of the cell contents on the cell wall is called as turgor pressure. The pressure exerted by the cell contents on the wall is called as turgor pressure. ठीक है. And the pressure exerted by the wall on the cell contents, it is wall pressure. भाई ये जो है ना, ये पूरे wall exerting pressure on the cell content me. So what is happening here? This is nothing else but this is wall pressure. I am exerting pressure on this. This is what is turgor pressure. ठीक है. If any time the cell wall is unable to bear the turgor pressure, it ruptures. मैं फोड़ दूंगी इसको, वॉल को, इस वॉल को. ठीक है. It ruptures and the cell contents burst out. This is exactly what happens when fruits and vegetables sometimes burst. When turgid, the cell is somewhat balanced state. No more water is entering or leaving it. Its turgor pressure counterbalances the wall pressure, and therefore there is no further absorption of water, even though the concentration of solutes inside the cell may be greater than the outside cell. Next देखो, it will shrink. कहाँ पर थी मैं यार, हाँ, the shrinkage from the cell wall, the shrinkage from the cell wall is called as plasmolysis, and the cells in the strate are called as flaccid, and the condition is called as flaccidity. Flaccidity is a reverse of turgidity. Okay, flaccidity is reverse of turgidity. Turgidity क्या है, मैं ऐसे ऐसे पुश कर रही हूँ, flaccidity is reverse of turgidity. ठीक है, flaccidity is reverse of turgidity. However, plasmolyzed cell is returned. Plasmolyzed क्या है, turgidity is the reverse, sorry, flaccidity is the reverse of turgidity. So however, if the plasmolyzed cell is returned to water before it dead, its protoplasm again swells up, pressing tight against the cell wall. The recovery of the reversal of the plasmolysis is called as deplasmolysis. You can easily understand that plasmolysis is the result of outflow of water from the cell, and deplasmolysis is a result of its re-entry. The terms defined, see, turgidity, plasmolysis, flaccidity. Turgidity is a state of a cell in which the cell wall is rigid and stretched by an increase in the volume of vacuoles due to the absorption of water. The cell is then said to be turgid. Plasmolysis is a contraction of the cytoplasm from the cell wall caused due to the withdrawal of water when placed in a strong hypertonic solution. Flaccidity is a condition in which the cell content is shrunken and cell is no more tight. The cell is then said to be flaccid. Uses of turgidity in plants. Turgidity provides rigidity to soft tissues such as leaves. Turgor pressure helps to push through the hard ground. Turgor in root cells builds up root pressure. Root pressure is a pressure developed in the roots due to continued inward movement of water through cell to cell osmosis, which helps in the ascent of cell sap upwards through the stem. Turgor in the opening and closing of stomata. Turgor movement. रूट प्रेशर अगेन ये भी है, एक्सपीरियंस ये बहुत इंपॉर्टेंट है एक्चुअली. एक्सपेरिमेंट्स आर वेरी इंपॉर्टेंट इन प्लांट फिजियोलॉजी. यहाँ पे मैंने देखा है बहुत इंपॉर्टेंट है. बट मैं करूँगी, मैंने कहा कि एट द एंड ऑफ द चैप्टर, नॉट एट द एंड ऑफ द प्लांट फिजियोलॉजी, एट द एंड ऑफ द चैप्टर आई विल डू एक्सपेरिमेंट सेपरेटली. ठीक है, अब लास्ट में एक बार पढ़ लेते हैं फोर्सेस कंट्रीब्यूटिंग टू एसेंट ऑफ सब, एंड देन विल मूव टू द एक्सपेरिमेंट्स. ठीक है, forces contributing to the ascent of soil: root pressure, capillarity, transpirational pull, adhesion. Root pressure builds up sufficient force to push the sap in the xylem vessels up to a certain height and may be enough for herbaceous plants. Capillarity or xylem vessels causes the water from a lower level to rise to fill up the vacuum created by the loss of water due to transpiration from the leaves. Narrow the diameter of the tube, greater will be the height of water rising in it. Exerting a force called as capillary force. Transpirational pull: as the water is lost from the leaf surface by transpiration, more water molecules are pulled up due to tendency of water molecules to remain joined. Cohesion, and thus to produce a continuous column of water through the stem. Addition, it causes the water to stick to the surface of the cells, thus drawing more water molecules from the below. When the leaf cell lose water during transpiration, this pulling force provided by the leaf is especially important in all trees, tall trees such as pines, which do not have enough root pressure. Downward movement of sap is relatively simple. The food manufactured in the leaves is dissolved in the water and it flows down mainly on the account of force of gravity. Cohesion is a molecular attraction of the particles of a body when united through a mass. Cohesion is a molecular attraction by which the particles of a body are united throughout the mass. Can you guess from where the भाई taste comes? पता नहीं भाई, मुझे नहीं पता है, मुझे मत पूछो ना. Okay guys, let's start with example. Experiment of diffusion, experiment of osmosis. ठीक है, experiment modified थोड़ा 2 वाला 3 वाला कुछ है नहीं वहाँ पे वैसे as such, but चलो यार पढ़ लेते हैं. मैं कर रही हूँ तो स्टडी कुछ नहीं करना तो पड़ेगा. Let me just, can I just sit like, I think I should just shift it. एक second. So much better than that because I don't know about you guys कि आपको कैसा लग रहा होगा, but trust me, I'm getting a back support and that's like Very big thing for me. So let's start now. Place a sugar cube or a small tablet of a soluble dye or a crystal of potassium permanganate in a beaker containing water in one corner. The sugar dye or potassium permanganate slowly dissolves and spreads in the liquid. Eventually the molecules of the substance are diffused or distributed uniformly throughout the water. In the movement of molecules there was no obstacle. There was no obstacle of any kind in the path of the molecules of the dye, and they could move freely in the water in all directions. Let us let us see how the identification how the definition of diffusion applies in this case. The molecules of the dye are more crowded, more concentrated in and near the tablet of the dye. These molecules move away further and further in the regions where they are fewer or absent. This movement continues until the molecules are uniformly distributed. Stirring with a spoon or a glass rod hastens the process of uniform diffusion, and you get a homogeneous solution much faster. Experiment number 2. See, take some concentrated sugar solution in a tissue funnel. Cover the mouth of a tissue funnel with a cellophane tape and tie it securely. Invert the tissue funnel in a beaker containing water and suspend it. So mark the level of the sugar concentration solution. The tissue set is set up. You just have to set up everything how they have showed us, and we will observe now. As a control, take another tissue funnel with a plain water filled in it and suspend it in another beaker also containing water. Again mark the level. After a few hours, the level of sugar solution in the tissue funnel experiment setup will rise. The level of sugar water in the tissue funnel is controlled will remain unchanged. Why? Again obviously भाई तूने उसको कुछ बांधा हुआ किससे बनाया है? वो क्या है? वो रबर से बांधा. रबर तो कुछ है नहीं, कुछ अंदर नहीं ना बाहर नहीं, राइट. तो वो रबर वाला इससे तो कुछ जाएगा भी नहीं, अंदर भी नहीं बाहर भी नहीं, बट दिस इज सेमी परमिएबल, सो इट विल लीव डिफ्यूजन ऑकर. सो देखो यहाँ पे द लेवल ऑफ वाटर इन द बीकर इन द एक्सपेरिमेंट विल ड्रॉप स्लाइटली व्हाइल वन इन द बीकर इज कंट्रोल्ड विल रिमेन अनचेंज्ड. इन इफ द वाटर in the beaker in the experimental setup is tasted, it is not found sweet. No, it's not found sweet. It's not found sweet. Okay, 3 main conclusions from this experiment we get. See, in the experiment setup, some water of the beaker has passed through the cellophane paper to enter the tissue funnel containing sugar solution. Sugar solution from the tissue funnel has not passed into the beaker. The cellophane paper has acted as a selectively or a differentially permeable membrane. It has allowed water molecules to pass through it, but not the sugar molecules. Experiment modified version of experiment, again इसका ही experiment 3 है, हमने 2 कर लिया है अभी. If you had slightly modified the experiment setup, taking a more concentrated sugar solution in the tissue funnel and very dilute or less concentrated sugar solution in the beaker, the result would be still same. The level of the sugar solution in the tissue funnel would rise. This again is because some water from the less concentrated sugar solution in the beaker would move in the more concentrated sugar solution in the tissue funnel by crossing through a cellophane paper. रबर शीट और मसलिन में अगर हमने करा होता वो कुछ होता ही नहीं, ठीक है. तो ये तो छोड़ ही दो, हम बेटर होगा. Two key points the above experiments. There are 2 liquids of different concentration. The sugar solution has a higher concentration of water molecules, of the sugar molecules, where they may be fewer or no sugar molecules in the beaker. It is very important to understand that the concentration can be visualized from the side of the solvent. There are more water molecules in a unit in the beaker than in the same volume of sugar solution in the tissue funnel in. Thus there are 2 regions of different concentration tonicity which you may express in the following: 2 regions of different concentration of water molecules, 2 regions of different concentration of sugar molecules. The 2 liquids are separated by a cellophane paper which behaves like a selective permeable membrane, a semi-permeable membrane in one which allows the passage of molecules selectively. It allows the solvent water molecules to pass through it freely but prevents the passage of solute sugar or salt solution. How long can osmosis occur? Theoretical osmosis should continue until the concentration of water molecules becomes equal isotonic on both the sides of the membrane. ठीक है. Osmotic pressure of a solution is a measure of its energy to take in water by osmosis. Is a measure of its energy to take in water by osmosis. Osmotic pressure in the experiment, an airtight piston bearing up some weight was introduced in the tissue funnel from the very beginning as shown. The level of the solution would not rise at all, showing thereby that there was no entry of water. Thus we may say that at osmotic pressure is equal to the weight or pressure required to nullify osmosis. So distal funnel piston level increased. So 1 second. Next we have I just hope my camera doesn't fall. Like I'm honestly just scared of that thing. तो अच्छे से रहे, ठीक है, बहुत इंपॉर्टेंट है वरना. Next we have root pressure के experiments. 1 second. Actually my grandma calling me to give me sweets, but it's okay. मैं वो और मैं लिटरली मैं पता मुझे गुस्सा आ रहा था कि क्यों भाई, वाई डिस्टर्बिंग माई स्टडी. बट yeah, next let's get back. Root pressure. While eating I was looking bad. बेड के ऊपर नहीं, यहाँ रखते हैं. गाइस मैं भी थिंकिंग दैट ये लड़की पढ़ नहीं रही है, बाद में पढ़ रही हूँ, ठीक है. मैं खाने पे ऊपर ध्यान नहीं है मेरा. I'm so insecure about my eating. Okay. मतलब नहीं यार, यहाँ पे देखो यार, मैं गंजी लग रही हूँ और मुझे और बाल खाड़ने की और मुझे इतना गंजा बनना है। नाटकी देखो मेरे, पहले से इतना बड़ा माथा है मेरा। Okay, a medium-sized young balsam plant is uprooted and washed and placed in a beaker containing a stain. A medium-sized young balsam plant is uprooted and washed and placed in a beaker containing a stain eosin solution, pink in color, pink in water. The roots should be completely submerged in solution. This setup is kept for 3 to 4 hours. At the end of this period, the plant is taken out of the solution and washed in. Experiment to show that water is conducted upwards through xylem. And tap water, there is a transfer section of root, stem and leaves is made and examined under a microscope. The xylem vessels will appear distant from the rest because these will be stained red by the dye. Conduction of water through xylem can be shown by another but delicate experiment. Take two leafy shoots such as those of a balsam plant which have been cut under water to prevent any air bubbles getting in, but keep their lower ends dipping in water. Remove 3 centimeter long outer ring phloem from the stem as shown in the beaker, keeping the central part intact. Ringing experiment, also called as welding in other beaker. Remove an equal length of the central part after incising the stem for full thickness and keeping the peripheral part intact. The shoots are then fixed to stands and are allowed to remain full. Guys, we are done. It took me half an hour to read one chapter. My half an hour, not half an hour at least, but some little is utilized. But yeah, now I'm gonna do a little fast studying of transpiration. Transpiration is the process of loss of water in the form of water vapor from the leaves and other aerial parts of the plant. Take a medium-sized well-watered potted plant, cover the plant with a transparent polythene bag and tie its mouth around the base of the stem. Leave the plant in sunlight for an hour or two. Drops of water will soon appear on the inner side of the bag due to the saturation of water vapor given out by the leaves. The water vapor condense only if the outside temperature is cool enough. The similar empty polythene bag with its mouth tied, kept in sunlight, will show no drops of water. If tested with dry cobalt chloride paper, the drops will be confirmed as water only if the blue paper turns pink. That is, there is a presence of moisture. There is a presence of what's this. So again, there's again experiment number 2. Take a small water plant, preferably one with broad leaves. हाँ, enclose the potted plant with a polythene bag, buy and tie. So arrange next वही सब कुछ अभी क्या होता है डायरेक्ट पढ़ो भाई, पता ही दिख ही रहा है. So at the first the bell jar A will show water condensing on the inner walls. The bell jar B will show a similar condensation. At the same time, the initially blue cobalt paper will turn pink. The blue cobalt color paper will turn pink. The third does not change. The third bell jar in this experiment is a control which provides that there was no moisture in the air due to transpiration, as there was no plant in it. By taking the 3 bell jars as described above, there is a double visual proof of transpiration, condensation of water vapor into droplets, and change in color of cobalt chloride paper. Measurement of transpiration, weighing method, and Ganon's photometer. ठीक है. A small light weighted potted plant can be weighed before and after the end of a certain period of time. The soil surface and the pot should be fully covered to prevent evaporation from the soil from the surface other than the plant. The loss in weight by plant during that time is due to loss of water by transpiration. An improvement in the weighing method can be made by using a glass bottle linked by a rubber tubule to a graduated side tube fitted with a water as shown. So the water level in the side tube falls, demonstrate water loss through transpiration through the leaves. ठीक है भाई, आगे बताओ. पोटोमीटर इज अ डिवाइस. तो हाँ, वेट, पोटोमीटर गई मैं. This would indicate the volume of water lost that can be compared with the loss in weight with the help of a weighing machine B or by covering into gums. Another weighing experiment can be done by using a test tube fitted with water and inserting a leafy shoot in it and pouring some oil on the surface to prevent loss of water from the test tube by evaporation. Place the test tube in a small beaker and wave them together. Remove the intact test tube and keep it straight in the test tube. Stand for few hours. Weigh it again, keeping the same beaker. Any difference in the weight will indicate loss of water by the shoot due to transpiration. Since there is no activity roots to activate water will be much less. So this is our weighing method. Potometers. This is potometer method. Potometer is a device that measures the rate of water intake by a plant and now first हम पढ़ेंगे. There are various type of potometers. तो आपको दिख रहा है मेरे बाल? It's something very bad. Something I don't know what. No, bad. Wow, bad. A twig of some suitable plant cut with a sharp knife is fixed in an apparatus as shown. The entire apparatus is fitted with water so that no water air spaces are present. An air bubble is introduced into the horizontal graduated capillary tube which is dipping into the beaker containing water. As transpiration proceeds, as the water is lost from the twig, a suction force is set up which pulls the water from the beaker and the bubble in the capillary moves along the rinding of the capillary tube with the volume of water lost in the given time. To repeat the experiment, the air bubbles can be brought back to its original position by releasing some more water from the reservoir into the capillary tube by opening the stopcock. Precautions in the use of potometer. The potometer should be made completely watertight. The twig should be cut obliquely to allow large surface for the water intake and under water to avoid suction of an air bubble into the twig which will stop the absorption of water into the xylem. Limitations in the use of potometer. Introducing the air bubble is not very easy. The twig may not remain fully alive for a long time. Any change in the outside air temperature may affect the position of the air bubble in the capillary tube. So this is how. Precautions are only the protometer should be completely watertight. The twig should be cut obliquely. Introducing air bubble is not very easy. Limitation: the twig may not remain alive for a long time. Any change in outside temperature will also affect it. Kinds of transpiration. Again, other has stomatal transpiration, cuticular transpiration, lenticular transpiration. Next, mechanism of stomatal transpiration, which is the first type of transpiration, are minute openings in the epidermal layer of the leaves. A stoma is surrounded by two bean-shaped guard cells. During daytime, the stomata are wide open, primarily for the intake of CO2 for photosynthesis. The number of stomata may range from 1000 to 10,000 per centimeter square. In stomatal transpiration, the water vapor escapes through the stomata of the leaves. Water, after the absorption of it by the roots from the water from the soil, rises up through the stem and reaches the tissues of the leaves through veins. A large number of spongy mesophyll cells in the leaves have their surface exposed to the intercellular spaces. These surface of the cells out some give out some of the water as a thin film. The water from this thin film evaporates, and the water vapor formed saturates the air in the intercellular spaces. The vapor then diffuses into the other connecting stomatal spaces. From where it escapes through the stomata. The entire movement of water vapor from the surface of the cell into the outside atmosphere is a result of diffusion. The molecules of water vapor, like those of any gas, move from region of higher concentration to the region of lower concentration. मैं क्यों अपने बाल पे हाथ लगाती हूँ literally? Very bad habit, very bad habit. बहुत गंदी चीज़ है, but yeah. Where it finally undergoes, the cell can lose water in this way. Replace it by drawing more of it from the nearest vein. Most of the water travels along the cell walls by imbibition, and only a small quantity enters the cell by osmosis. Thousands of leaf cells evaporate water in this way, causing more of water to be pulled from the below via the xylem vessels. The transpirational pull thus created can draw up water to up to 50 meters or more in tall trees. More transpiration occurs from the under surface of a dicot leaf. There are more stomatal openings in the under surface of a dicot leaf, and therefore more transpiration occurs from the under surface. This can be provided by an experiment in which pieces of tricobalt copper are attached to two surfaces of the leaf and held in position by two glass lights on either side together by elastic bag and are held by clips. The leaf should remain attached to its own place. So up and down, so more stomata are held down. So that is what stomatal regulation. Transpiration: stomatas are minute structures occurring in large number on lower epidermal of the leaf. Transpiration occurs as long the stomata are open, but it stops when they are closed. The opening and closing mechanism of stomata is regulated by the amount of water and cellulose present in the guard cells. As the stomata are open during the daytime, the diffusion of gases in and out starts fulfilling the need for photosynthesis as well as allowing transpiration. If for any reason the water content of the leaf is falling short, the guard cells fail to remain turgid, rather they turn flaccid, thereby closing the stomatal opening and transpiration stops. A more detailed account of opening and closing mechanism of the stomata on the basis of potassium ion exchange theory is given in the next chapter. ये भी पढ़ना था, ये भी पढ़ना था. Wow, my little bit. ठीक है. Leaves of some plants wilt during midday and recover in the evening. In some plants blossom, the leaves of the plants wilt during the midday in the spite of the fact that there is plenty of water in the soil. In such cases, the rate of transpiration during midday exceeds the rate of absorption of water by the roots. The cells therefore lose turgidity in the evening or during the night. The stomata are constricted and the temperature is not high, therefore there is no loss of water through transpiration, and the turgidity of leaves is reacquired and they stand out erect. Stomata transpiration is controlled by plants by adjusting the size of stomata, whereas this does not happen in case of cuticular and lenticular transpiration. Cuticular is a waxy layer secreted by the epidermis on the two surfaces of the leaf. The thickness of the cuticle varies from plant to plant. Primarily, the cuticle serves to prevent evaporation of water from the leaf surfaces. However, some evaporation does occur. The greater the thickness of the cuticle, the lesser evaporation. Desert plants tend to have thicker cuticles to cut down transpiration. Lenticels are special openings that develop on the barks of the old stems. These allow diffusion of gases for respiration. Lenticels never close, they remain open all the time. Water from the cell surfaces directly facing the lenticel evaporates and contributes to the transpiration. The amount of transpiration from the cuticle is certainly much less than the stomata transpiration but more than the lenticular transpiration. Factors affecting the transpiration: light intensity of light, temperature, velocity of wind, humidity, carbon dioxide, atmospheric pressure. Internal factor: water content of the leaf, adaptation in plants to reduce excessive transpiration. Sunken stomata, fewer stomata, narrow leaves, reduce exposed surfaces, loss of leaves, thick cuticle. Significance of transpiration: cooling effect, suction force, distribution of water and mineral salts, distribution of water and mineral salts. So that is all you have to remember. Adaptation in plants to reduce excessive transpiration: you can have fewer stomata, narrow leaves, reduce exposed thick cuticle, and loss of leaves, sunken stomata. ठीक है ना. अगेन नेक्स्ट में क्या आता है, एक्सटर्नल फैक्टर्स दैट अफेक्ट ट्रांसपिरेशन: लाइट इंटेंसिटी, टेम्परेचर, वेलोसिटी, विंड वेलोसिटी ऑफ विंड, ह्यूमिडिटी, कार्बन डाइऑक्साइड, एटमॉस्फेरिक प्रेशर, ऑल दिस. सो सिग्निफिकेंस क्या है? कूलिंग इफेक्ट, सक्शन फोर्स, डिस्ट्रीब्यूशन ऑफ वाटर एंड मिनरल्स. ट्रांसपिरेशन विच क्लाइमेट, इट हैज बीन शोन experimentally that plants give out large quantity of water during transpiration. A full grown single sunflower plant is estimated to lose about half a liter of water per day in the form of water vapor. A single maize plant loses 2 liter. An apple loses 30 liter. So forests contribute in bringing rain. Transpiration is the secret. But now the secret is known to everyone. Next, guttation and bleeding. Some plants may lose water or other fluids along with dissolved substances directly in liquid form and not as water vapor. This is known as exudation. To exude means ooze or sweat out, and fluid given out is known as exudate. It occurs in two ways. Guttation. The leaves of certain plants exhibit droplets of water along their margins in the morning. This particularly happens in plants growing in warm, humid conditions. A humid environment hampers transpiration while the roots continue to absorb water from the soil. This builds up a big hydrostatic pressure within the plant and forces out the excess water directly from the tips of the veins in the leaf. Special pore-bearing structures called hydathoids are present on the margins of the leaves to allow the exudation. Guttation can be observed in many plants. Is quite in banana, Nasturtium, strawberry bleeding, Nasturtium. Bleeding this happens only due to injury in the plant escapes from the rupture of the rupture or the cut. Swimming up. Guttation, bleeding. It occurs on the edge of the leaves by the hydathoids. It occurs in the injured plant. Exudate mainly plant sap, sugar. It occurs time of injury. Early morning, early night. Exudate mainly water and minerals. This is plant sap and sugar. It occurs in all the plants that have been cut or injured. It happens in certain plants like banana, nasturtium, strawberry. It happens in all the plants. It occurs in like very few plants. Guttation occurs. Example एक जो वो है हमारा स्ट्रॉबेरी और बनाना, याद रख लेना. Next we have photosynthesis. ठीक है, and after photosynthesis we have chemical coordination. I just want to read chemical coordination पहले so that it's just small and we can just finish it off, and we can have like a nice time for photosynthesis because photosynthesis is really very high weightage as well as important. So let's read chemical coordination. वैसे ज़्यादा क्वेश्चंस बिल्कुल नहीं, ज़्यादा क्वेश्चंस क्या भाई, दो चार भी ज़्यादा हो गया यार. दो क्वेश्चंस आते हैं सिर्फ इस पूरे चैप्टर्स में से, बट ठीक है पढ़ लेते हैं अब. I'll just have a 5-minute break. Done with the break. Let's start. So yeah, oxygen. The term oxygen was first coined by F.W. Wendt in 1928 from the Greek word ox auxin, which means to grow. It is the first growth hormone discovered in plants. Auxin are powerful growth stimulants and are quite effective at extremely low concentrations. Auxins are universally distributed in higher plants as well as in lower plants like algae, fungi, etc. In higher plants, they are present more in actively growing regions like shoots, apex, roots, apex, lateral meristem, etc. IAAA, indole 3-acetate, is the main natural auxin found in animals. Functions of auxins. Auxins promote the growth of the stem, roots, fruits by cell elongation. During this process, cell wall is extensively increased. This hormone induces cell wall loosening. For relaxation, plant cell wall gets stretched like a rubber when put under tension. Rate of cell elongation is proportional to the auxin concentration. Stem cell elongation requires more auxin in comparison to roots. Auxin delay leaf senescence, aging or falling. Auxin promote the growth of apical buds and inhibit the growth of lateral buds. In majority of vascular plants, apical buds dominate over lateral buds. The growth of the lateral buds occur only after the removal of the apical buds. This phenomenon of the suppression of the growth of the lateral buds by apical buds is called as apical dominance. Auxins induce rooting in cutting of some plants like rose, bougainvillea, etc. Auxins can induce fruit formation without fertilization in fruits like apples, tomatoes, bananas, etc. Such development of fruits without fertilization is called as parthenography, and the fruits are called as parthenographic fruits. Gibberellins are another kind of a plant hormone. There are different forms of gibberellins reported from different plants named GA1, GA2, GA3 and so on. However, gibberellin acid GA3 is the most studied form. In higher plants, gibberellins are mainly disturbed in meristem regions like stem apex, root apex, buds, seeds, etc. The main function of the gibberellin is to promote the growth of the internodes by the cell elongation. Gibberellins break seed dormancy and initiate germination. They promote fruit growth and are capable of inducing parthenography. They also delay senescence, aging. Gibberellins are also widely used in horticulture and food industries. These hormones enhance longitudinal growth of internodes in dwarf plants. They are used commercially to increase the length of the grapes, elongate apples and improve their shape. Gibberellic acid is used to speed up the malting process in brewing industry. Cytokinesis. The cytokinesis have been discovered comparatively recently in 1950s by Shung and Miller. Cytokinesis have specific effects on cell division. Cytokinesis are widely distributed in plants. They are produced in root tips and are transported through xylem cells. Comparatively large amounts of cytokinesis are found in germinating seeds, developing fruits and embryos, etc. Cytokinins stimulate plant growth by cell division as against auxin and gibberellins, which stimulate growth by the cell elongation. They promote cell division even in non-meristematic tissues. In seeds, cytokinins cause expansion of the cotyledons. They break seed dormancy and promote germination. They promote chlorophyll synthesis in chloroplast and delay leaf senescence. Cytokinins inhibit apical dominance. Ethylene is the only hormone which is a gas at ordinary room temperature. It is produced in fruits and remain in the same fruit. Therefore, unlike other hormones, its site of synthesis and site of action are not different. A gas causing ripening of fruit was first found to be emitting from oranges, which help in the ripening of bananas when stored together. The gas was then found to be ethylene by R. Gane. Ethylene is produced in higher plants and fungi. All living cells are capable of producing ethylene, but more ethylene is produced in meristem tissues. Functions of ethylene. Ethylene causes two processes in plants: reduction in stem elongation and acceleration of senescence, ripening in fruit, initiating germination in peanut seeds, sprouting of potato tuber, promoting root growth and root formation, inducing flowering in mango, inducing formation in mango. Thus, ethylene is the most widely used plant growth hormone in agriculture. APA, abscisic acid. Abscisic acid is a growth retarding hormone. It is found in angiosperm, gymnosperm, heterosperm, and some